This is Mike Yeoman with Mature Science News, bringing you the top stories from the 1860s era. First up, Are You My Mommy? In 1855, Rudolf Virchow published a paper that claimed that all cells come from other cells, even though he had initially thought the division only occurs in certain types. There have been allegations that Virchow was actually publishing the work of Robert Remak. It Takes Two. Charles Lyell and Joseph Dalton Hooker arranged for both Charles Darwin and Alfred Wallace to present their theories jointly in 1858. Darwin continued to work on his future best-selling book on the origin of species, while Wallace continued to research the importance of biogeography to natural selection. No Respect is the story of Gregor Mendel's life. He published a paper in 1866 which demonstrated the predictable expression of visible traits and coined the terms dominant and recessive. This paper eventually became the foundation of modern genetics. Talk Wordy to Me. In 1861, Paul Broca provided evidence for the localization of brain function. He performed autopsies to show the presence of lesions on a particular part of the left frontal lobe in patients who suffered from aphasia, a language disorder caused by damage to the brain. This location is still known as the Broca area, and many of Broca's patients' brains are still preserved in France. Can you hear me now? In 1863, Hermann von Helmholtz postulated the theory of hearing by examining the ear as a sensory organ. This led him to the resonance theory, where certain frequencies vibrate definite locations of the cochlea. His work also led to the measuring of the speed of nerve impulses. These colors don't run, they flow, because they're your blood. In their groundbreaking 1862 and 1864 discoveries, Felix Hopf-Seyler and George Stokes discovered the function of hemoglobin. Hopf-Seyler described the absorption spectrum of the blood pigment, while Stokes saw a change in color from red to purple to red again when exposing the hemoglobin to a reducing agent, then to the air. We are not alone. There is something living inside of your cells. Between 1850 and 1888, Albert von Kolliker first identified the mitochondrion in muscle cells and eventually separated them from the rest of the cell structure. This feat showed that mitochondria contained their own plasma membrane. Only the good die young. Otto Dieters was good at science and died at the age of 29 from typhoid fever. But before his passing, he extensively used microscopes to study the brain and spinal cord. He discovered the axon and dendrites of neurons, providing the current general image of the neuron that we use today. Dieters postulated that dendrites must fuse together to form a continuous network. As the Friends character Ross Geller said, nobody likes change, except for Claude Bernard, who shortly before 1868 supported the concept that we now know as homeostasis. He wrote, "The stability of the internal environment is a condition for the free and independent life." Bernard asserted that a large part of this ability to maintain an internal environment was due to the fluid circulating in the body. He was also the first to suggest blind studies in order to prevent bias in research. Stop that bleeding right now. That's what Max Schultz might have said, the first person to accurately describe the platelets in blood. He was researching the effect of temperature on blood cells, mainly white blood cells, and found these recurring spherules that seemed to be a normal constituent of his blood and other animals. This paper, published in 1865, was 44 pages long, without a single subheading. The burn means it's working. Dr. Joseph Lister published a series of articles in 1867 after using carbolic acid as an antiseptic. Lister changed the practice of medicine by introducing physicians to washing their hands with 5% carbolic acid solutions and making medical instruments out of non-porous materials. Lister got the idea from reading a paper written by the French chemist Louis Pasteur regarding fermentation and the spoilage of food. Which brings us to our main story tonight, spontaneous generation. Fermentation and bacterial life in general were perceived as being spontaneous phenomena. Louis Pasteur knew that his germ theory would not be fully accepted as long as belief in spontaneous generation continued. Pasteur used a simple experiment to address the problem. Beef broth could be sterilized by boiling it in a swan neck flask that has a long bending neck that traps dust and particles before they reach the broth. When a second flask was boiled and the neck broken off afterwards, the broth would be exposed to the air and turn cloudy. The cloudiness indicated microbial growth, proving that life does not generate spontaneously. A huge discovery for the 19th century. Thanks, and now it's Jack with sports.